Good evening, everybody. My name is Christian Kujo, an MPhil medical microbiology student in the University of Ghana. I am going to take you through the concept of viruses today. When you look around in your environment, in your house, in your rooms, anywhere you find yourself, you are being surrounded with a number of microorganisms. And these microorganisms affect us in every aspect of our daily lives. You would think that microorganisms only infect humans, but microorganisms do not only infect humans. When you look at these diagrams are what? Microorganisms. This is a virus, this is a bacteria, this is a fungi, these are protozoans. Previously, before the inventions of microscope, these life forms were previously not seen with the naked eye. When you look at here, we have a structure of a virus. Here, this is a virus. We are having virus here. This is a bacteria. This is a fungi. We're having protozoans. In fact, we're having a number of microorganisms that we cannot see. The fortunate thing is that the most fortunate thing that we have as humans is that these microorganisms, most of them are not harmful. In fact, some of them are not infectious. They don't cause human diseases. I think you have not forgotten the nitrogen-fixing bacteria, Nitrobacter. You were taught this one when we were at the basic school. And you have also not forgotten Saccharomyces cerevisiae. That is the baker's yeast, the one that we use in bread baking. We also use some of these microorganisms for production of drugs, for example, penicillin. So some are beneficial and others also causes human diseases. So let us go. Today we are going to consider when you got up this morning, you have mentioned virus because of coronavirus. Every day when you wake up from your bed, you mention the word virus. Viruses do not only infect humans, they also infect plants. So we have plant viruses, we have insect viruses. And one interesting thing is that viruses also infect bacteria. Viruses are microscopic, bacteria are also microscopic. But we have viruses that infect bacteria, bacteriophage. So the diagram you see here is a bacteriophage virus. Back at school, those of us who study science, we are familiar with this structure. This is a bacterial virus, a virus that infects bacteria. We have animal viruses, that is virus that be the humans. Example is the coronavirus, the human immune virus. Let us consider the structure of a virus. How do we identify? How are we able to see that this is actually a virus? A virus is an obligate intracellular pathogen that has an outer capsid, that's a protein coat, and an inner core of nucleic acids. So you just look at something here. Look at the cursor. The structure you see here is the capsid. The outside protein coat is the capsid. They are made up of individual protein subunits called capsomes. I'll talk about the capsomes later. Within the capsid are nucleic acids. The nucleic acid can either be RNA or DNA. A particular virus doesn't have both. It doesn't have both RNA and DNA. It may have only one, either DNA or RNA. In fact, they are too small to be seen by the light microscope. The microscope that we often have in our labs. When you go to the local labs, we're having these light microscopes. Unfortunately, these microscopes cannot be used to visualize viruses. A virus is 20, the size ranges from 20 to 300 nanometers. Nanometer is very small. When you take 1 meter rule, you divide 1 meter by 1 million, it give you 1 meter. So The parvovirus, the picornavirus, the smallest virus is the picornavirus, ranges from 20 nanometers to 24 nanometers. To the biggest virus, that ranges up to 300 nanometers. Let's go to the structure of a virus. When you take a typical virus, I have told you that the outer protein coats and an inner core of nucleic acids. The outer protein coat is made up of what? Protein subunits. A virus may come in three forms. You're going to get three forms. You will get either the one in icosahedral shape, the one in helical shape, and the one that is complex. It is neither icosahedral or neither is also complex. When you take the icosahedral shape, why are we going to see that this virus has an icosahedral shape? This individual protein subunit called the capsomes, you can count 1, 2, 3, 4, 5, 6, 7 in this order. These are capsomes. In some viruses, the capsomes are arranged. They are arranged in equilateral triangle. Let us look at the pointer from here to here, down here to this part will give you a triangle. You can also start from here, go this way, come this way, and you also get a triangle. 20 of such triangles will come together to give you an icosahedron. Singular is icosahedron and plural is icosahedral. We have other viruses that are helical. With the helical virus, the capsomes, these are the capsomes. The capsomes are attached to the nucleic acid, that is the DNA or RNA. This is the RNA. Once they are attached, as the nucleic acid is winding, twisting, then the structure is also twisting. In that order, we will get the forming helix. You get a structure forming helix. And these are the helical viruses. We have other group that are complex. They don't have this icosahedral shape, and they don't also have this helical shape. We call them as complex viruses. So basically, we have three major structure of a virus. A virus will come as icosahedral, will come as a helical, or will come as complex. As we go down, we'll be able to know some of the helical viruses, the icosahedral viruses, and those stuffs. Now you can watch this diagram. I have explained that a typical virus will have a capsid, a nucleic acid. This is the general structure of a virus. But in some viruses, they also have something we call the envelope. The envelope surrounds the capsid. This envelope is conferred on the virus during the final stages of replication. We also talk about replication. Human beings give birth to their young ones. Virus also replicates. They make copies of themselves inside the host cell. So after the copy has been made, it will come out of the host cell to infect new cells. During budding, when they are budding out of the host cell, they are coming out of the host cell. You know, human cells, I'm using humans because we are humans. So when a virus infects human cell, it enters into the cytoplasm. It means that it will have to cross the membranes, the cell membrane, into the cytoplasm. In some with some viruses, they have to even cross the nuclear membranes and get into the nucleus. So after replication, when they are coming out of the cell to infect new cells, they would tear some of the nuclear membranes of the host cell to surround themselves. And these viruses are called enveloped viruses. So the envelope is obtained from the host cell, and it is not coded for by the virus. They don't get it from the virus, but they get it from the host cell. Corona is a typical example. Coronavirus has this envelope, and the major function of this envelope is to protect this capsid and also protect the nucleic acids. It also gives a structure. It gives a structure to the virus, and it also confers antigenicity. When I say antigenicity, I will explain later. So just let's focus. So now we also have naked viruses. Those that do not have the envelope, they are called the naked viruses, and those that have the envelope are called enveloped viruses. So now we know that we are having an envelope virus, we have naked viruses, we are also having icosahedra, we have what we even call it, helix, and we are having the complex. This is just a diagrammatic thing to show you how the virus obtains the envelope. Let us take this, Let us take this to be the cytoplasm of a host cell, here to be a cytoplasm of a host cell. Now, after replication, it will need to cross the nuclear membrane. So this is the membrane. Here is the membrane. When it crosses this membrane, it will have to tear this membrane and surround itself with. This will give us what? The envelope. So we have an envelope. The envelope is made up of phospholipid, a certain fatty fats. They are made up of certain fats, phospholipid, and also glycoproteins. So the spikes, these spikes you see surrounding the virus is what? A glycoprotein. Unfortunately, these glycoproteins are coded for by the virus. It means the virus will have to produce this glycoproteins by itself. But the phospholipid is found in the membranes that the virus obtained from the host cell. So what we have studied, we are having 5 major groups of virus. We are having the naked icosahedra virus. When we say a virus is naked, I think by now you get the concept. A virus is naked and icosahedral. It means that it doesn't have the envelope around itself. It has that icosahedral shape, the one that I said earlier. So a virus will also be enveloped and icosahedral. A virus can be naked and helical. A virus can be enveloped and helical, and a complex virus. So we're having 5 major groups. Now, how is the envelope medically important? Why do we even need to know something about the envelope? Why would some virus obtain envelopes from their host cell? We are taking this is an HIV, a human immune virus, and this is a host cell, CD4 cell. Now, when you look here, the virus will have to attach to the host cell. This is a virus, and it's attaching to the host cell. Now, every virus will have this receptor at their cell surfaces. They will be having this receptor. These are receptors. And the function of this receptor is to bind to the host cell. You see, this is binding to the host cell. This is binding to the host cell. In human immune virus, we are having the GP120 and GP41. That binds to the host cell. The host cell receptor is CCR5 and it's also having a co-receptor. So it will need to bind to the host cell before it enters the cell. It is this envelope that is giving the virus opportunity to do so. So in medicine, if we are able to block, we get medicines to block these receptors. Then it means that the virus cannot even bind to our host cell. We have a drug called azidothymidine. This azidothymidine is an HIV drug. Its function is to block these receptors. So when they block these receptors, the virus cannot bind. It will not come and attach and then cause infection. Another important thing of how is the envelope medically important is that some of the chemicals we use, for example, the detergents that we use, will also cause these receptors, damage to these receptors, and then they will not be able to bind to the host cell. Let's go forward. We're talking about heat. How is the envelope medically important? We are talking about heat and cold. We heard that, oh, we have to be heating our food. Some even say you have to inhale vapor. You have to inhale vapor so that you get rid of corona. Some viruses are actually not too comfortable with heat. These enveloped viruses are not comfortable with heat. We say they are heat labile. When organism is heat labile or a virus is heat labile, it means that it doesn't tolerate heat. So heat at 37 degrees Celsius for some time would actually deactivate the virus. So it is actually true. We can use heat to get these enveloped viruses deactivated. But when you talk about cold, you don't freeze. You don't put your food into a freezer and think that, oh, the virus has been deactivated. A virus, an enveloped virus can live, can stay within your fridge even at a temperature of minus 90 degrees Celsius for a prolonged time. So we don't actually use freezing to control enveloped viruses. And let's go forward. Ether and detergent. The common detergents we use in our homes for cleaning and other purposes also do have a negative effect on viruses. In fact, I have explained the enveloped viruses to you. The detergent will have effect on the phospholipids, that's the phospholipids present in the cell membranes, that's the membranes that were derived from the host cell. Detergents will have effect. It will cause it to solubilize, that it becomes soluble, and it also have effect on the capsid. You know, I told you that the triangles are joined together, isn't it? They are joined together. These ionic detergents, the ones that we have been using in our homes, will prevent these triangles from joining. So it will separate them, it will put them into separate groups. And when that is done, it exposes the DNA. And once the DNA is exposed, it means that the virus can be deactivated or its infectivity will be lost. With alcohol and what, that is what we have been using, alcohols. Use alcohol hand wash sanitizers to deactivate virus. Use it to wash your hands, use it to wash surfaces so that you get the virus deactivated. It is true. There is a Casapreco hand sanitizer. It contains alcohol, either isopropanol or ethanol. And this isopropanol or ethanol will also have the same effect. It will react with the lipids in the cell membranes. Just that I told you, corona is an enveloped virus. So it will react, this alcohol, that's the ethanol, will react with the phospholipids in the cell membranes of the corona, that is the envelope, and cause it to be what? Deactivated. When you are sick, you don't go to the hospital. You just go to the drugstore to buy a drug. The most drugs we have in the drugstore are mostly antimicrobial agents. When I say antimicrobial, I mean they are effective against bacteria. Those drugs are not effective against viruses. So you go and take any sulfonamides, or you take penicillin, you take any drug. If it is antibiotic, I must assure you that it doesn't have, those drugs doesn't have any effect on viruses, so you don't use them as such. When this corona came, I saw that they were spraying outside with chlorine. It is true. High concentrations of chlorine also have negative effects on viruses. So you can use high concentrations of chlorine to deactivate viruses. Now that we know the outer structure, features of viruses, it is important that we get inside what is there in the, when you cross the envelope and you cross the capsid. When you cross the capsid, we are having, I told you earlier that a virus may be having either DNA or RNA. So with the nucleic acids, a virus will have either double-stranded DNA. So the DNA will be double-stranded as the one you see here. You see there is one strand and there's another strand that is winding together. So we are having a double-stranded DNA. And we also have a single-stranded DNA. So a virus will also be single-stranded DNA. If it's an RNA, you can also have a single-stranded RNA, and you can also have a double-stranded RNA. So here we are getting 4. Double-stranded DNA, single-stranded DNA, single-stranded RNA, and double-stranded RNA, 4. The RNA can be segmented or non-segmented. When you say segment, it can be in this way. You get it? This here, it is in segments. You can remove this segment out. You can remove this segment out. So some of the nucleic acids, either DNA or RNA, can be segmented or non-segmented. It can be circular this way. You see, this one is a circular. It's in the form of a circle. And this one is linear. So it can also be linear. And it could also have positive sense, or it will be negative sense. We will understand this one better when we are treating replication in viruses. Because if a virus, the reason why some viruses will have a short incubation period, that's when they enter into the host cell. About 3 or 4 days, you develop symptoms. And in some cases, it will be there for a long time, 6 months, no symptoms. It has something to do with the positive and the negative senses. So we'll talk about that one later. After describing the nucleic acids, we have other important enzymes that are also present there in the virus. We have something we call the polymerase. It is an important enzyme during replication. And we are also having the reverse transcriptase. This reverse transcriptase is very important enzyme to the human immune virus. So when we are describing pathogenesis of human immune virus, we are going to make mention of this enzyme, reverse transcriptase. And we're also having an integrase. It is also an enzyme. It's also a very important enzyme to the human immune virus. You've heard that it is difficult to treat infections from human immune virus. And it is true, partly because of this integrate enzyme. It does something. When we get there, you know its activities. And we also have the protease enzyme. Now, per our understanding of viruses, are we going to say a virus is a living thing, or we say virus is a non-living thing? You have a reason to say virus is a living thing. When you say virus is a living thing, it means that the virus is able to undergo all the 7 life processes. And if you're saying it is not a living thing, it means that it does not undergo all these 7 life processes. I think we are aware of these 7 life processes. We were taught back long way back at school, GHS or SHS. Now, based on our knowledge of either a virus being naked, being enveloped, having single-stranded DNA, double-stranded RNA on all those stuff, we use this principle to group viruses into what? Families, and even into genera. Based on what? The type of nucleic acid. Now, when we say nucleic acids, I think we know what nucleic acids are. Size and morphology, that is either helical, either complex, either what and what and what. And the mode and site of replication, we'll talk about that one too. And presence of these enzymes, either DNA or RNA polymerase. Based on this, we are having we have so many group of viruses. We are having the parvovirus. Example of this parvovirus. Remember, the most important member of the parvovirus is what? Human parvovirus B19. We are having papoviruses. You have heard of papilloma virus. We also have a group called the herpes viruses. It is where the varicella zoster is. Epstein-Barr virus, cytomegalovirus. We are having HepaDNA virus. These viruses include the hepatitis B virus. We are having the pox virus. This is the cowpox and the monkeypox. So all these viruses, all this family of viruses contains DNA. So these are DNA viruses. So when you hear hepatitis B virus, you know what a DNA is. It is a DNA virus. And other features we will describe later when we consider hepatitis B virus alone. All these, the stuffs listed here are families of RNA viruses. In fact, most of these pathogenic viruses are mostly RNA. So you start from here, you end here, which includes the almighty coronavirus, the one that is battling humans now, coronavirus. They're having the important members are the SARS-CoV, the SARS-CoV-2, and then the MERS. These are all this thing of the coronavirus. Now, with our knowledge, what we have learned today, this is a structure of the coronavirus. Is it an enveloped virus? What is here? These structures here I have explained these structures. What is it? If you are able to identify this structure, you are able to tell us that, oh, this is an icosahedral shape, this is an enveloped virus, this is that, then we are good to go to the next section of biology. Next time when we meet, we are going to take the viruses one by one, but I would want us to consider vaccines first. Next week on Chrisvee Medical Lectures, we'll be talking about vaccines, because the information we are getting is that the information we are getting is that people are saying they won't go in for the vaccine when it is ready, because they think it will cause what. So next week we will talk about vaccines into details, and we will know if coronaviruses, we have a vaccine for coronavirus, are we going in for or we are not going in for. So thank you, and I'll hand over to the program host to continue. Thank you. If you have any question, I think you can ask me. And
okay. Hello.
hey, hi, please, I can hear you.
Hi.